our last video lecture of Bio 130. So we're going to go over Chapter 16, the second part of the reproductive system, covering fertilization, pregnancy, and development. We're going to conclude with different types of contraceptives. As we left off last time, we looked at oogenesis and the ovarian cycle, and we saw how a surge of estrogen from this growing and developing Graafian follicle will then increase and incite the anterior pituitary gland to secrete luteinizing hormone. This in turn is going to cause the secondary oocyte to ovulate. Now, where exactly does the secondary oocyte enter? It enters into the peritoneal cavity. Meaning that here you see the ovary, and here's that secondary oocyte that's been ovulated. If you notice, it actually this secondary oocyte, more often than not, will enter here into our fallopian tube. And that's because we have cilia lining this fallopian tube that's going to create this nice pull of and wave-like motions of the fluid within this fallopian tube to pull that secondary oocyte towards this fallopian tube. However, sometimes the secondary oocyte may not even enter into this fallopian tube, in which case it can land right outside of this fallopian tube, and this can give rise to what's called ectopic pregnancies, in which case if the sperm ends up outside and you can fertilize the egg, you can have the maturation of that embryo to that fetus outside of the womb. So kind of scary and very dangerous for that to happen. However, I'm going to clear this off really quick. We're going to go over normal circumstances when that secondary oocyte does enter into that fallopian tube. So here it is, and as it waits for the sperm to come, the sperm's going to come into this region over here. So let's get started with fertilization. This oocyte, that secondary oocyte that has just been ovulated, is viable only for 12 to 24 hours after ovulation. And once again, that one oocyte, that one that's mature the fastest, is going to be one that's going to be ovulated. So there's only one viable oocyte after oogenesis from one ovary. Sperm, on the other hand, are viable for 24 to 48 hours after ejaculation, and what's pretty crazy is that 1,500 sperm are produced per second. Now, I'm not a math major or anything, but if you look at the calculation, 1,500 sperm per second times 60 seconds per minute times 60 minutes per hour times 24 hours a day gives you 129 million sperm that's being produced a day. Crazy, right? Now, for fertilization to occur, sexual intercourse must occur no more than 2 days before ovulation, because we need the sperm to be viable, and no later than 24 hours after ovulation, because once again, those oocytes, secondary oocytes, are only viable for 24 hours after ovulation. Now, it's not an easy task for all the sperm to get towards that secondary oocyte. And to give you an idea, it's kind of like a big race. What is that called? The Amazing Race. So here, it's the Great Sperm Race, where you have 250 million competitors and one winner. And of course, the winner gets to fertilize the egg. Now, because the sperm have to travel so far, they have to go from your seminiferous tubules within your testes to your epididymis to your ductus deferens, out of this ejaculatory duct, through that urethra of the male system, and then finally into the female. More than 99% of that sperm will be dead or dying, and after between 1 to 2 hours, it will take sperm to reach that uterine tube, so a very long time. Now let's take a look at some of the factors that can cause the sperm to die. So we took all the time just to get through that male reproductive system. Now if you get into the female's reproductive system, that's a whole nother story. Vaginal canal. We have a very acidic environment, so a lot of the sperm is going to die off within this area. Cervix, cervical canal right over here, very narrow, so also it's going to take a long time for all that sperm to get through. A lot of it is also going to get stuck within this canal. When it enters into the uterus, it doesn't meet the obstacles don't even stop there. You have all these macrophages. They're going to try to engulf all of these foreign sperm, like, oh, what is this going in my body? I'm going to attack it. It's not something that's not known to me. This kind of reminds me of The Purge, if you guys have watched The Purge. Anyway, moving on. And then you got to go through this fallopian tube, this long raceway to get to that egg. But by the time it enters into this fallopian tube, As it nears the egg itself releases chemical signals that drives and draws its sperm closer. So pretty much near that last leg, as it approaches the egg, it's pretty much chilling. It's kind of nicely going towards that egg. And then finally, of course, only one sperm is going to fertilize that egg. Now, once that sperm comes into contact with that egg, it's going to undergo what's called an acrosomal reaction. Remember what the acrosome looks like. Here's that head, we have the midpiece, and then we have the tail. And we have that helmet, kind of like that warrior helmet on top of that sperm. And this warrior helmet, the acrosome, contains digestive enzymes, hyaluronidase enzymes. And what these enzymes are going to do, it's going to break down the corona radiata around that secondary oocyte. Here's that secondary oocyte, and remember we have this corona radiata right around it. So these enzymes are going to break down this corona radiata so that the head of the sperm can interact with the egg itself. Now, once it comes into contact, the very first sperm to come into contact and break through this corona radiata and make contact with the membrane of the secondary oocyte is going to be that sperm that's going to enter. All that DNA, genetic material found here within this head, is going to be the one that's going to enter into the egg. Let's look at these events one more time. So here we have this acrosomal reaction, in which case we release all those digestive enzymes, hyaluronidase enzymes, into this space where we can start digesting away this corona radiata. Digest through this membrane, and then this first sperm, this is the first sperm that makes it through, this genetic material, all this DNA, is going to fuse with the plasma membrane and enter into that cell. Now, the moment that that membrane of that sperm meets with the female's membrane of the oocyte, a second sperm head cannot enter. So it's going to actually create this protective shield that's going to surround around, let's see here. So it's going to make this layer much harder and prevent any further penetration from any sperm trying to enter into that egg. The moment that it comes into contact, that secondary oocyte comes into contact with the genetic material of that sperm, it's going to undergo the second meiotic division to form that ovum and a polar body. Fertilization will then be completed once that genetic material of that sperm combines with that of an oocyte. And what do we result with? We result with a zygote. So let's see what that looks like. Okay, so prior to that sperm arriving, we have the secondary oocyte. This is the one that's found within that fallopian tube. So we are ending over here at the end of meiosis 1. Remember, this has half the genetic material, and this other has half of that other genetic material. Upon fertilization by that sperm, if you recall from the last video lecture, I'm just going to draw what this looks like over here. We had one copy here, and we have the other copy here. This copy is found within that secondary oocyte. This copy here is found within that first polar body that we see over here. Once that sperm comes into contact with this egg, half of this genetic material is going to start fusing with the sperm. So this is going to split. Blue. And then it's all by chance. It could be either this genetic material or this genetic material that's going to fuse with this incoming sperm's genetic material. And remember, this has 23 chromosomes. This one contains 23 chromosomes as well. And the moment they come together, now you have your 46 chromosomes, which forms is a zygote. It's diploid. And a zygote is that fertilized egg. And over time, this is going to develop into an embryo and then develop into a fetus, in which case that fetus is the form that you take on when you are born. Here are a couple of videos for fertilization. I'll also post this in LaLima for you to watch. Now, the zygote, this is a term we give to this new cell that has just been formed by the fusion of both this DNA from the sperm and that egg. And the moment this occurs, we're going to undergo a series of mitotic cell divisions. And this is going to, as the cell continues to grow and divide, not necessarily grow, but it's going to continue to divide, it's going to move down this uterine tube towards that uterus. Here is this fused zygote containing genetic material from both your mother and father. And we're just going to take this exact same type of egg and continue to split it. So we're not making it grow, we're just dividing that to create more surface area. Okay, and this is, as this continues, it's going to go down the entire uterine tube. A couple of terms that we need to know as we continue the pregnancy and development. Pregnancy is a time from fertilization until the infant is born. Conceptus is a term that we give to that developing offspring, so different terminology for this. From fertilization until week 8, we call that developing offspring an embryo. And during this entire phase, we have lots of maturation of the internal organs and development of those different organs. However, by week 9, a lot of these organs are already developed, in which case we start calling that offspring a fetus. And we call it a fetus from week 9 until birth. Gestation period is a time from that last period until birth. So this is about 280 days. Here's a diagram illustrating the sizes of a human conceptus from fertilization until the early fetal stage. And of course, here we have this cell. It's going to undergo a series of mitotic divisions and cell divisions. And then by the 3rd week, we call it an embryo. It's going to start developing the different layers that are going to give rise to either our nervous system, our muscles, our skeletal system, different organs and systems within our body. Here's a 5-week embryo, 8-week embryo, and then here's a 12-week embryo at 90 millimeters. We're now going to take a closer look at cleavage, being that series of mitotic divisions, beginning with that fertilized egg, being that zygote, and ending with a blastocyst, which is a form that zygote takes on as it's implanted into that uterus. 24 hours after fertilization, that zygote's going to start to divide, first in half, and then in quarters, in eighths, and then in sixteenths. The moment that it divides into 16 cells, we call it a morula. But that division does not stop there. We're going to continue mitotic division of this exact same type of cell mass until it forms to resemble something that looks like a gumball machine filled with gumballs. And so we form this solid mass of cells, and each of these cells that we form inside, these are called blastomeres. At this point, it has been 3 to 4 days after ovulation, and this stage of the morula is going to enter into the uterus. However, it will not be implanted into the uterus just yet. When that ball-like circle of cells reaches 100 cells, we give it the term a blastocyst. And this blastocyst will secrete the hormone, human chorionic gonadotropin hormone, which we saw last time, HCG. And this is going to cause that corpus luteum to continue to produce progesterone. Remember, we need this progesterone to maintain that endometrium. We have 2 regions of this blastocyst. We have a trophoblast. Here's what this blastocyst looks like. We have a area here. This is all filled with fluid. This is that trophoblast region. And we have this inner cell mass composed of cells all located to this one side. Let's do a quick overview of the steps of fertilization and cleavage, starting off with this ovulation of the secondary oocyte from the ovary into that peritoneal cavity, and then finally the secondary oocyte will enter into the fallopian tube, where it's going to await fertilization from sperm. The moment sperm fertilizes this egg, it's going to form into a polar body and an ovum. This ovum that's now fertilized is called a zygote. And the zygote is undergoing cell division 24 hours after that fertilization. It'll form a 4-cell stage body 2 days after fertilization, and then 3 days after, it's going to form a solid ball of blastomeres called a morula. We had that initially with that 16-cell stage, and then culminating in this massive ball of cells. This morula is not going to be implanted into the uterus just yet. It'll be free-floating until it develops into a blastocyst. And this blastocyst is going to have 2 major regions. We have a trophoblast, which is this area containing a fluid-filled cavity, and an inner cell mass over here. And this form is going to be the form that is going to implant into that uterus. In the past, I've had questions of how are fraternal twins and identical twins formed and developed. And I'm going to quickly draw the ovaries, fallopian tubes, uterus, and then here's the vagina. Now, prior to fertilization, sperm will enter both fallopian tubes. It does not know which side. It's pretty much a 50-50 chance of where is that secondary oocyte going to be found. Now, two ways you can get fraternal twins. One way is if the ovary does release two mature secondary oocytes into the fallopian tube, in which case the sperm does fertilize each, and that's how you can get fraternal twins. So fraternal twins, these will not look identical, they will not look exactly alike, and that's because they are both from separate eggs and also from separate sperm. And so with fraternal twins, you can have one being a male and also one being a female. Another way you can get fraternal twins is by having one secondary oocyte being released by one ovary and another secondary oocyte being released by another ovary, in which case sperm, of course, half of them are going to go this way, half of them are going to go their way, so both of them are winners. Yay! And of course, this one can also result in both having a male or a female. Identical twins are different in that the formation of both of these embryos are from one egg and one sperm. And it all occurs at this morula stage, in which case the morula can spontaneously divide to form two separate maturing embryos, in which case each of these embryos are going to have the same placenta, however separate amniotic sacs, and these are going to be identical. So identical because they have all that same genetic material that they first initially started off with. However, identical twins cannot be both male and female. It can only be one gender. Let's take a step back and look at the importance of this blastocyst in that it forms the primary germ layers that give rise to our major organ systems. We have three primary germ layers, the ectoderm, mesoderm, and the endoderm. Ecto, meaning outside, gives rise to our epidermis, hair, nails, lens of our eye, our retina, and as well as our nervous system. Endo, meaning within or inside. This is going to be the epithelial lining. Anytime you see mucous membranes, whether it's going to be in our alimentary canal or also within the region going towards our respiratory tracts. Parts of our liver, pancreas, bladder lining, and our thyroid. And then finally, everything between our endoderm and our ectoderm is going to be our mesoderm. So everything between these alimentary canals and respiratory regions, and our ectoderm being our skin, we can think of as our muscles, our skeletal system, those blood vessels, excretory organs being your kidneys, your reproductive structures, your lots of connective tissues, and portions of your digestive and respiratory system. By day 14th, after that fertilization, our blastocyst will implant into that uterus. Here's an illustration of those three primary germ layers, the ectoderm in blue, mesoderm in red, and the endoderm in yellow. And it's a good idea to, once again, go over these primary germ layers and what they give rise to. Development after implantation. So if we look at that blastocyst, that trophoblast region of the blastocyst, this will eventually develop into the chorionic villi, which is going to cooperate and combine with cells of the uterus to form the placenta. And this placenta forms a barrier between that mother and the fetus, and developing fetus. If you recall, there's only one antibody that can cross from that mother to the fetus, and what was that? That IgG. And that prevents any cross-immunoreactivity between that mother and fetus. However, of course, which we're going to get into, there are other things that can cross from that mother to that fetus, and embryo being alcohol and drugs, so affecting development. In here within this placenta, we have this fluid-filled sac surrounding the embryo to prevent it from shock, and that's being the amnion. And connecting that placenta to the embryo is the umbilical cord, which gives rise to all the nutrients and oxygen for that fetus. Okay. Here's this placenta. Here are the chorionic villi that are going to extend into the uterus. Here we have the amniotic sac, fluid-filled sac, umbilical cord, and of course umbilical vein being that high nutrient, high oxygen source for that developing embryo. Let's look at these functions of the placenta, forming that barrier between that mother and embryo where that blood is not exchanged. We can deliver nutrients and oxygens by that third week of fertilization, as well as remove waste products from that embryo through the blood. It also becomes an endocrine organ by taking over for that corpus luteum to maintain that inner lining of that endometrium by that second month by producing estrogen and progesterone. Remember, both of these also needed for that mordant and that basic building block of that entire endometrium, and also to maintain pregnancy. Another role and function of this progesterone, which is very important, is it also helps to reduce the amount of contractions of this endometrium. Now we're transitioning from an embryo to a fetus beginning the 9th week. And why is that? Because all of our organ systems are formed by the end of the 8th week. And so a lot of the activities of the fetus are for growth and organ specialization. And so we're going to start changing appearance of the fetus by the 9th week. And by 270 days after fertilization, the fetus is going to be full term and ready to be born. Here's an illustration of that fetus, month 3, and later month 5. So lots of that is just development and maturation. Of course, we can't do anything about developing this embryo to this fetus without the mother. And the mother herself has to undergo lots of anatomical and physiological changes to respond to this developing embryo and fetus. First thing is enlargement of the uterus, in which case, as the embryo develops to that fetus, we also need to account for more room and more space. And then to account for more of this growing embryo to the fetus, we also bear more weight more anteriorly, and therefore the mother's curvature, to account and to allow herself to balance, we're going to have an accentuation of this lumbar curvature. Other things, we have relaxation, widening, and flexibility of the pelvic ligaments to prepare for the labor. And pubic symphysis also becomes more relaxed and more flexible due to the hormone relaxin. So just think of, what does relaxin do? It relaxes the pubic symphysis. If you recall where that pubic symphysis is, here we have the oxcoxyl bones, and then combining them here, or allowing them to connect in the front, is this ligament right here, and that's a pubic symphysis. Before conception, the uterus of the size is about size of a fist, so it's right within that pelvis. 4 months, the fundus is this broad area of the uterus. It's halfway between the pubic symphysis and the umbilicus. 7 months, fundus is above that umbilicus. And 9 months, the fundus reaches the xiphoid process. And if you remember, that's part of the lower region of that sternum. It will extend into there. So of course, we can have, because we have that barrier, blood barrier between that mother and the fetus through the placenta, we can also allow different things whenever the mom ingests into that embryo or to that fetus, which can cause birth defects and even death. So alcohol, nicotine, and drugs all can cross from that mother into that fetus. Let's look at the physiological changes that occur on the mother. First, we have the gastrointestinal system. Let's just take a quick look over here. So as we recall, as that fetus starts developing, we have all those other abdominal organs that it's putting pressure on. So we have our small, our large intestines, our stomach, pancreas, spleen, liver, all those internal abdominal organs. And so because of that, we can have a couple of changes. Heartburn, when that overcrowding of that fetus puts pressure on your stomach. We can have those gastric juices go towards that esophagus, giving rise to that heartburn sensation. Morning sickness, this occurs within the first few months of pregnancy due to elevated levels of progesterone and estrogen. And constipation, you remember your large intestine, it's going to undergo mobility and peristalsis every couple of hours. And because we have pressure now from this developing fetus, we're going to put pressure on that large intestine and reduce the mobility and motility of that digestive tract. Urinary system. Not only is the mother taking in more water in order to account for the growth and development of the cells of the embryo and the fetus, but we also have to get rid of waste products from that offspring as well. So we have to produce more urine and more metabolic wastes. Also, this developing embryo or fetus is going to be compressing that bladder even more, making urination more frequent, urgent, and also uncontrollable. Respiratory system. Okay, so we saw earlier that the fundus of the growing fetus is going to start putting pressure on that xiphoid process, which in turn can also put pressure along the diaphragm and that respiratory system itself. And because of that, we decrease the amount of oxygen in and out of our lungs, so you will increase your respiratory rate, and you'll also need to increase your vital capacity to compensate for the decreased amount of oxygen in and out of your body. Also becomes more difficult to breathe during these later stages of pregnancy, which we give the term dyspnea. More changes! Oh my goodness. Thank goodness for all of you who are mothers. We don't appreciate how much you put your body through for those 9 months. Let's look at cardiovascular system changes. Total body weight and water levels rise because we have additional needs for that fetus. Blood volume increases 25 to 40%. Also, if we look at the pressure from that developing embryo and fetus on our pelvic blood vessels, this is going to reduce the ability for venous return from that lower limbs, giving rise to varicose veins, more pressure of the lower limbs, and also edema. Okay, so of course, for those of you who are mothers, you can tell more stories of all the other pains and different symptoms and signs that you've gone through, and for that we have much respect for you. Let's now switch over to childbirth, which we also call parturition, and this is a culmination of pregnancy within 15 days of the calculated due date, and that is 280 days from the last menstrual period. And a series of events that expel the infant from their uterus we give the term labor. And this will be a rhythmic, explosive contractions that's operated by a positive feedback mechanism. And remember, this is only the one of 2 positive feedback mechanisms we have in our body, in which case we want to increase the stimulus and push that stimulus further. Labor is initiated by increasing levels of estrogen during the last few weeks of pregnancy, which is going to cause the uterus to start having weak contractions, and we term this Braxton-Hicks contractions. This is called false labor because we are not yet giving birth to the fetus. It's just contracting the entire uterus. However, these weak contractions are going to start stimulating the cells within the fetus to produce oxytocin. Oxytocin is going to cause the placenta to release prostaglandins, which causes the uterus itself to contract. Both these weak uterine contractions and further contractions from the uterus will cause physical and emotional stress on the mother, which causes the mother's hypothalamus to release oxytocin by the posterior pituitary gland. Now, the oxytocin from the posterior pituitary gland and prostaglandins together will give rise to true labor, because now we're truly causing that uterus to contract. And once again, all of this will occur through this positive feedback mechanism, and we'll see in a little bit how this actually works. Let's go over one more time the events of early labor until finally we have labor. And we initiate this by prostaglandins and Braxton-Hicks contractions that put pressure against this uterus to force the baby downwards. This head of the baby is going to put pressure against the uterus, and also the stress itself is going to stimulate the mother to send signals towards the hypothalamus to release oxytocin from that posterior pituitary gland. The oxytocin itself is going to target the mother's uterine muscles to cause contractions. Once that happens, contraction of the uterus again is going to continue to push the baby down even further against the cervix of the uterus, which in turn is going to continue to send more stimulus to this afferent neuron towards our brain. Brain and the hypothalamus will stimulate the posterior pituitary gland to release oxytocin and continue to repeat the cycle. And so every time that baby's head puts more pressure along the surface, the stimulus will continue until finally that baby is expelled. There are 3 stages of labor. We have the dilation stage, expulsion, and placental stage. During dilation, this is when the uterine walls begin to contract and the contractions are increased. The infant's head is forced against the cervix, and the cervix wall itself are going to be thin and soft to allow for the passageway of the fetus. During this time, the amniotic sac also ruptures, which gives rise to that water bag breaking, and the cervix begins to dilate. This is the longest stage of labor, being 6 to 12 hours. During expulsion, we have full dilation at 10 centimeters, in which case we need enough room for the infant's head to pass. Remember, you recall from the skeletal system, the infant's head is the largest region of the entire body at birth. And this expulsion stage can last as long as 2 hours, But typically, on average for the first birth, it's about 50 minutes, and the second birth and thereafter, it should be around 20 minutes. Normally, the head is first to allow oxygen into the fetus. However, sometimes the butt can come out first, in which we call it the breech presentation. This can offer some difficulties, especially when you want to allow air to enter into that fetus's circulatory system. And in some situations, if the fetus is not head first, we can also give rise to a cesarean or a C-section, in which case there will be an incision through the abdominal wall and also the uterine wall. What I've seen before, I actually did rotations at a hospital for as a biomedical intern, and I saw a woman getting a cesarean, and it was one of the most disgusting things I've seen. Not because the procedure is disgusting, but it's just the way that the doctors handled the situation. So what they did is they made an incision about, I think it was about 4 inches long, across the abdominal wall and also through the uterine wall. And both of the doctors were standing. So if this is an incision, here's the skin, here's one flap of skin, and here's the other flap of skin. They were grabbing their hands along each wall of the skin of the abdominal and uterine wall, and they were leaning back to rip the mother open, and it was horrifying. There was blood everywhere. And I was asking, well, what is the purpose of this? And they were saying that when you have cesareans, and if anytime you have a large area that you need to cut through, it's better to make an incision that has a rigid edge so that when it heals, they can kind of come together like puzzle pieces and interlock, versus having and creating an incision that's 10 inches long, for example, just with a sharp edge, in which case it's going to be more difficult for these edges to adhere together. But yeah, pretty gruesome. Okay, third stage of labor is the placental stage. A placental stage is when we deliver the placenta, and it's accomplished within 15 minutes after birth. And so it's going to be contraction of the uterus that's going to continue after this birth. It's going to compress down along the placenta, and then eventually this placenta will detach from the uterine wall. And this entire contents from this placenta and the umbilical cord, as well as any residual amniotic fluid within this cavity, will then be released into the external environment, and that's called afterbirth. Okay, then looking at developmental aspects of the reproductive system, how do we determine gender? And sometimes you've seen some families where they have nothing but all daughters and female cousins, or all males, but actually that's all by chance. And really, the sex of the child is all determined by the male. So if you want a guy or if you want a girl, it's all dependent on your husband or your boyfriend. So here is we'll just show, this is called a Punnett square, and this can show the probability of you having a male or a female. So the top XX denotes female chromosomes, and XY denotes male chromosomes. And so every time you have a child, you take 1 and 1 from the male and the female. Here we have XY and then XY. So all in all, how many XXs do we have? We have 2. And how many XYs do we have? We also have 2. So really, anytime you have a child, it's a 50-50 chance of having a boy or a girl. And once again, it's all determined by the male, because the male carries that Y chromosome, which will determine whether or not your fetus will be a boy or a girl. Gonads themselves do not begin to form until the 8th week, and what triggers the development of these female or male structures is the amount of testosterone, meaning that at birth we have our genetic makeup of either being male or female. However, we actually both start off as females, and then over time, depending on this testosterone, we're going to start developing either male or female structures. So here we have pseudohermaphrodites and hermaphrodites. And this is when we have interference with a normal pattern of sex hormone production. And so pseudohermaphrodites, these are individuals who possess external genitalia that does not match their own gonads. If you have embryonic testes that don't produce testosterone, the males will produce female accessory structures and external genitalia. If females are exposed to testosterone as they grow and develop, the embryo will have ovaries, but will also develop male accessory ducts, glands, the penis, and an empty scrotum. Hermaphrodites are individuals who possess both ovarian and testicular tissues. They typically need sex change operations and, of course, cannot fertilize themselves. Hermaphrodites are individuals who possess both ovarian and testicular tissues, and in which case they typically get sex change operations and they cannot self-fertilize. Let's take a quick look at the developmental aspects of the reproductive system. Males sign onset of puberty when they have enlargement of testes and the scrotum. This occurs around age 13. And females will start their puberty often around the age of 11, and we know this by the physical appearance of budding breasts. And this is a term that we learned from last video lecture in menarche. This is a first menstrual period in a female. Other developmental aspects of the reproductive system: menopause. This occurs when a whole year has passed without menstruation, and the ovaries stop functioning as an endocrine gland, meaning that the reproductive organs in response and also the breast tissues will start atrophying or dying. The vagina becomes dry, irritable. There's more vasodilation of the skin's blood vessels, giving rise to hot flashes. And also thinning of the skin and loss of bone mass are also common. During this phase, your childbearing ability also ends. There is no equivalent of menopause in men. Remember that in men, they can continue to produce sperm until a very old age, even 94 years old, if you are very determined. But there is a steady decline in testosterone. Now, there's one more thing that I do want to introduce before I get into contraception, and that is this lady who actually she was an older lady. She went to the hospital because she's having abdominal pain, and they found out that it was a calcified fetus that was within her abdominal cavity. And I'll show you the article and the video on that on Malima, but I just thought it was pretty crazy. This lady, she was actually in the process of giving birth, and she saw someone who was giving birth die on the table. So she ran out of the hospital, and she was experiencing so much abdominal pain, but she kind of just ignored it. And over time, because the fetus was considered foreign to her body, the body itself started forming this calcium enclosure around this fetus. And so for so many years, I think it was 40 years, she had this calcified fetus within her until she finally had it removed how many years later. So I'll send you that article and you can watch the video. It's pretty gnarly. Okay, our last topic for our Bio 130 class is contraception, and this is looking at birth control and preventing pregnancy. Birth control, the most used contraceptive is the birth control pill. As it stops ovulation, and we went through this last time when we looked at the synthetic forms, because estrogen, synthetic estrogen, and synthetic progesterone, these are not going to make the conditions hospitable for the endometrium and implantation for that embryo and that fertilized egg. It also helps to maintain estrogen levels so that we don't have a peak in estrogen, therefore we don't have a peak in luteinizing hormone, and therefore we don't have ovulation. Other types of contraception, we have sterilization, tubal ligation. This is cauterizing or cutting the uterine tubes. Of course, you can also tie your tubes, so cutting or cauterizing these. If you tie them, you can prevent fertilization temporarily. However, once you untie them, then you can go back to returning to having babies. Vasectomy, this is cutting or cauterizing the ductus deferens, and this occurs closest towards the testes. So it's cutting close towards this region and prevents its sperm to enter into that vas deferens. Coitus interruptus, this is when you withdraw the fetus prior to ejaculation. And you can also look at your calendar to see when your female will ovulate, and you can avoid intercourse during those periods of ovulation. And so if we look at this hormonal regions of when ovulation will occur, you remember the sperm is viable within 48 hours, so we don't want to do any intercourse with the ejaculation of sperm 48 hours before ovulation, and then of course 48 hours afterwards, just to have that nice gap. And then within these days, these are considered more safer areas, in which case that egg will not be fertilized. But of course, the main thing is also barrier methods, in which case you can prevent any sexually transmitted diseases. Diaphragms, cervical caps, sponges, condoms, spermicidal foams, and gels. All prevent skin-to-skin contact, as well as decrease the mobility and kill off sperm cells. Abortion, this is termination of pregnancy, and a miscarriage is a spontaneous abortion that is common and frequently occurs before a woman knows she is pregnant. And how the abortion pill works, the RU486, this is going to block progesterone's quieting effects. Remember, you need progesterone to maintain that endometrium, and if we don't have that progesterone, then what's going to happen? This is going to be really unstable, and therefore that'll eventually give rise to that abortion. Here's an overview of the different techniques of how you can prevent the formation and fertilization and occurring. So you can take a look at this as an overview. And before we end this chapter, I just want to bring up two more topics that I think are very important, and that's the sexually transmitted diseases that are prevalent here in Hawaii. And specifically, we have two, chlamydia and gonorrhea. And I think it's important for us to understand this and to understand the community that we live in, especially if you are sexually active and you don't use protective means or barriers if you do have intercourse. And I think it's also important, especially if you know younger people who are 15 to 24 years of age. So here's a study, chlamydia, what is it? It's a sexually transmitted diseases. If a woman is pregnant with an infection, it can also pass that to a baby during childbirth. This can get into your eyes, it can get into your mouth. Chlamydia is a bacteria. 75% of women are asymptomatic. 50% of men are also asymptomatic. And what are some of the characteristics? Well, you can have vaginal discharge, bleeding, pain during urination. For men, clear, cloudy discharge from around the tip of the penis, and also pain and swelling around the testicle. Now, here you see it also in the oral cavity. So for those of you who are active, also think about using oral barriers between your oral cavity and external genitalia. Second one is gonorrhea, also another type of common STD. Also occurs with unprotected anal and vaginal or oral sex. And same thing again, pregnant women can pass on these infections to a baby during childbirth. So here's an instance or situation in which case if there is a bacteria within the vagina or vaginal canal, it can enter into the eye of the baby. So very important. Remember, just be aware, be cautious, get tested. If you have friends who are sexually active, make sure that they get tested as well. Other signs, burning sensation when urinating, discharge from the genitalia, whether it's white or yellow or green, and also bleeding in females. So these are a fairly recent report, January 2013. So just something to be aware of yourself and also for the others in your community. So once again, I leave you off with this illustration and also a reminder to use protection if you are sexually active. And that's it for Bio 130. Yay, you're done! It was a pleasure getting to read all of your bios and communicating with all of you through email. I wish you the best of luck in all of your future endeavors. And I will be posting your final grades by next Friday. Aloha!